Hello, students. Welcome to my 3rd out of 4 screencasts for this test coming up. This one will be on cell membranes and transport. And here we are at Trampo Extreme right here in Muscat, Oman. And once you go to these places and you've learned about diffusion and transport, it's hard to look at all these balls and not think about them diffusing somewhere. It's pretty funny. And then here's a hallway, right? And so making just some examples of getting you around to your different classes at TASM would be an example of transport. All right. Once again, I hope that you can see this connected to the other parts of the test, such as homeostasis, right? This is all about homeostasis, or getting internal conditions constant and or restoring them, and dealing with the cell cycle and cancer and cells in general. All right, let's dive right in. So you can pause the video and see if you can do these 9 questions about cellular membranes and transport. All right, how are you feeling? You feeling really prepared? If so, kudos to you. If not, go through this video and re-go through the 2 Pogles I gave you on it. Remember, I jammed the both of them together in classroom. I believe in the Pogles. I think they're really good, and they have those key questions there with them. And a lot of teachers use them, and I think they're very helpful. But don't just do, a Pogle for it. You also need to make your own models, and we did that in class, and we did that with the dialysis tubing bags. We did it with a lab. Hopefully that really helped you. We did it with oil and food coloring, and some of you did it with Mr. C, dealing with water coming in and out of plant cells. And if you didn't do it with him, you will do it with me. All right, let's jump right in. Okay, so here is a phospholipid bilayer membrane, right? And this is in all of our cells. It also makes up the membranes of the organelles within the cell. So this is really important. All right, do you remember that the head is hydrophilic? It is a phosphate. That means it likes water. So it goes towards the outside of the cell, towards the outside water, or towards the inside water of the cell, right? Over 70% of our bodies are water. The tails are lipids, and they don't like water. They're hydrophobic. So they fear water. They face towards each other, and they make this zone in here that is non-polar, students. So it's going to repel anything that's polar. It's going to repel water as well. So here we have a pretty cool membrane. Right? And these membranes is another example of emergent properties that lets us compartmentalize things and do stuff that's greater than if they're just by themselves. Okay, can you describe the fluid mosaic model? So here, this is a cool GIF. It's just showing, right, that it's not just static. The things are moving around. Cells tend to move. Here you can see stuff coming in and out. So I really like it. Here you can see that non-polar interior. Here would be some cholesterol. You think of cholesterol is bad, but it does help our cell membranes. Maybe it has markers. So this is saying this is a certain type of cell. The most famous markers you might know about are your blood type markers. So which items can easily cross the membrane? So things that are non-polar can pass right through the membrane. And they need to be small too, right? So things like carbon dioxide and oxygen, et cetera, they can just pass right through. Things that are polar, like sugar or water, et cetera, cannot just pass right through. Okay, so here we go. If they can't pass through the membrane, so let's say that they're polar, they're charged. It could be a sugar molecule, it could be like Na plus, like a cation, or it could be an anion. Well, how does it get into the cell? They need to use protein channels. And so this is pretty cool, right? If you remember, proteins have R groups. Well, the R groups that are nonpolar, facing the outside right here, anchoring it within the cell membrane. And then on the inside of the protein, like right around here, will be some polar R groups that help polar molecules go through. It's another beautiful example of how the structure of a molecule can lead to its function. So there's going to be different channels for different items that need to get in and out of the cell. Right? So here would be a sugar channel. It's called a GLUT4 receptor. Here's one for amino acids. Here's one for water. That's called an aquaporin. The Nobel Prize is given out for that right when I graduated from college. Here's one for salt and for waste. Okay, this is a flow chart I got from Bozeman Science. And so I've posted his video as well. It's excellent. Going through active and passive transport. Can you pause the video here and kind of describe what's going on? Okay, how'd you do? Hopefully all these words look familiar to you. We did do a worksheet on it in class. I'm trying to not do many worksheets. I want you to learn through investigations, etc. But I did find it helpful to just kind of review these terms. All right, and hopefully you see why it's important to learn about this stuff, right? Did we learn about some diseases going on here? Okay, so here's diffusion going from an area of high to low concentration. That is called passive transport, and it does not require any energy. Like an example would be the diffusion of water. This isn't entirely accurate, right? They'd be going through little pores called aquaporins. Here, well, where is it high and where is it low? Hopefully you can see that it's high on the outside of the cell and low on the inside, so the fat's going to move in. Here, high outside, low inside, so that sugar is going to move into the cell. Active transport is when we need to go against that gradient. So here, this is from the amoeba sisters. You can see it going from low to high, right? There's more of them on this side, so they would tend to flow this way, but we need to get them out. The best example of this is the sodium-potassium pump, right? We're getting all the sodiums out of the cell so that they're ready for an action potential. And here it is. Here go the sodiums out. We see here's ATP, adenosine triphosphate, so it drops off a phosphate. That gives energy to this pump, and that pump's going to kick 3 sodiums out and bring 2 potassiums in. All right. And why was that important? It dealt with action potentials and with nerve cells, et cetera. I'm not going to go through this slide and give you an in-depth talk about it. I hope you found it cool. We did those models in class. The point was just to show you the action potential, or show you the sodium potassium pump in action. All right. If you want to learn more about that, take AP Bio with me. Okay, hopefully you should be able to do this. Describe which way the water will flow. So this is from your study guide. Let's start over here. 75% water, 25% solute, 45% water, 55% solute. Well, it's going to go from an area of high to low concentration for water. So it's going to go from here to here. So it's going to go into the cell. That's going to cause it to swell. This is called a hypotonic solution. It makes it swell up like a hippo. Here, 90% water, 63% water, so it's going to leave the cell. The cell will shrink, and that is called hypertonic solution. It's hypertonic relative to the cell. Here, the water will flow in and out at around equal rates, and that is called an isotonic solution, and it is at dynamic equilibrium. So the easiest way I find for 9th grade students to think about this is going from high to low concentrations of water. If you want to think in a different way, if you only knew the solutes, right, it's going to go towards a higher solute, right? It's because the water would go around them and surround them. But let's just stick from high concentration of water to low concentration. So these are from Kim Foglia, so thank you for these slides. To give credit where credit is due. So the water is going to come into the cell. This is hypotonic relative to the cell. An example of this would be a paramecium, and that's why they have contractile vacuoles. It's not a problem for plant cells because they have a cell wall that can push back on the water. Here's an example for hypertonic, right? So the cell's losing water. For example, shellfish, they're going to have to figure out some way to take up water. Right, so this would be like salt water out here. So like when you go in the ocean and your fingers get all wrinkled up. And then the right amount is isotonic, such as our blood. So you watched the video clip of the young athlete dying from hyponatremia when the brain swelled because he drank way too much water after exercise. We also watched the holding the wee for a wee thing. So those are just some real examples of transport. Why do we spray distilled water on vegetables inside of a grocery store? It's to keep them looking fresh, just like you saw there. And here's the contractile vacuoles for paramecium. Can you figure out why do you gargle salt water when your throat is sick? Hopefully you can, and hopefully you can use some of these terms to describe it. All right, students, keep on rolling.